I am Molly. People call me Molly. And I joined the center in 2009. Before I joined the center, actually, just like you, I was a bench scientist working in the Heart and Lung Research Institute. If you have a question about my lecture later on, you can always email me or call me. The license you are having right now will last for like 1 week. So please take the advantage of this period. So if you have a question, just email me. I'll be happy to answer your questions. Okay, this is the kind of summary of what we had for this workshop. In day 2 yesterday, Dr. Fernandez actually gave a lecture about how to design the experiment for the RNA-seq experiment. Once you send your samples to the core lab, they sequence your sample, did alignment and quantification. After that, the data have to be pre-processed, like filter out those genes with very low expression. And also a lot of time, like for example, the TCGA consortium, they offer the normalized data, which you can use right away, so you don't have to normalize that. But a lot of time, you can also have the count data that you need to do the normalization. And I think in day 1, Dr. Brook gave a lecture of how to download the data from the TCGA website. And also there's another big respiratory for the public available data set that's in the NIH GEO site. After you download the data will be through univariate analysis and multivariate analysis. And yesterday, Mathieu and Greg went through how to test the differential gene expression and using the GLM to do the analysis. For the multivariate analysis, Dr. Coombs talked about the clustering yesterday and classification today. And Dr. Yu talked about how to predict the genes which are correlated with the overall survival by using a Cox regression model with different methods. And now you have a nice results, having a comparison result with the fold change or difference with the FDR or raw p-values. What can you do with the results? Usually you can see thousands of genes got upregulated or downregulated. There are many methods to summarize the data, the results you have. One useful tool is the Ingenuity pathway analysis. Actually, it just tells you which pathways or networks are involved, are regulated in your data set. Here's the outline of my lecture. First, I'm going to talk about what is Ingenuity pathway analysis. And I'll give you an example about the chronic myeloid leukemia. And that's the GSE number is the GEO accession number. So whenever you type the GEO number, the data will pop out. And later, I'm going to give you the application examples, including how to use IPA to help you to write a good manuscript, and how the IPA can help you to do the hypothesis generating. I think because of this computer doesn't have the IPA installed, so I combine the demo and the self-practice. I give you the instruction of how to upload the data into the IPA and how to do the basic analysis by using the tool. What is IPA? Actually, IPA is a web-based bioinformatics tools for quickly analyzing and interpreting the biological meaning in omics data. I think earlier on we talked about clustering and classification and prediction. They're all based on the data you have. But right now, a lot of people really want to know what happened in other people's research, what conclusions they have, and which can help you to generate hypotheses for your own research. And yeah, this is the website of the IPA. I think everybody had got the email yesterday about how to install the IPA on your computer. What is IPA? Actually, IPA contains a big database. It's called Ingenuity Knowledge Base. The company actually, they abstracted a lot of information from published literatures and conference abstracts, a lot of patents, and also from the database, for example, the target scan, which you used for finding the microRNA targets. And also they extract the information from the FDA, the drug information, and the KEGG website. That's used, a lot of people use that to find the pathways. So they borrow a lot of information from other websites too. Here give you the summary how the IPA works. Actually for IPA, you input genes. Actually right now they have the metabolomics analysis in the IPA. So also have the molecules, metabolites. You can also input into the IPA. As I say that you can input the public information from the TCGA or from the NIH GEO. And also you can input your own data, like it can be messenger RNA, microRNA, and proteins, and metabolites. And then you upload the data into the IPA, and then the IPA will output pathway analysis and disease and function analysis results, and networks, upstream analysis, and regulators. Here is actually it's a brief summary of IPA. This is how it looks like when you open the IPA. So I think the apoptosis signaling, it's very popular. That's always the first page of the IPA. And okay, I think here, when you look at that, you can see the IPA not only can analyze your data, you can also use it as a search engine. You can input the gene names, and chemicals for some drugs names, and diseases and function. For example, if you type the lung cancer, it will give you a lot of information about lung cancers. And also, if you are interested in DNA repair pathways, just input DNA repair pathways. After you click this tab, it will give you a lot of information. The search engine actually can save you a lot of time. Because a lot of time you try to use the PubMed. It gives you so much information, but this is more relevant to what you are searching. When you open the IPA, you try to do, for example, new core analysis. There's a window will pop out. It will tell you how to do that. So always use this workflow to help you to analyze your data. Whenever you have questions, you can go to the support. There's a support here. And in the support link, there's a flexible, the frequent asked question and a lot of things. You can get a lot of information from the support. And also you can call them to help you. So now I'm giving you an example. As I said, that's the go to the GEO website. If you know the study, you're interested, GSE number, just type the accession number. It will give you the data, expression data or other type of data you are asking for. Right now, most of the journals require you to deposit your data into the GEO website before you are able to submit the journal, submit the manuscript to the journal. So I think they try to use available resources for people because high-dimensional data analysis can be very expensive. So that's try to use the resource efficiently. In this study, there are 3 groups of patients. They are pH positive, Philadelphia positive, chronic myeloid leukemia patients, either at accelerated stage or chronic phases. And they also included like probably 8 normal donors. Usually you cannot get a lot of normal donors, usually more patient number than normal donors. There are 3 comparisons you can do. There are many comparisons you can do, I think 3 here. And here is the paper. And so I uploaded the data. I downloaded from the GEO website, the expression level data, gene level data. I uploaded the data into the IPA and analyzed that. Once the analysis has been done, the company will send you an email and tell you it's done. You can also wait in front of the computer, but a lot of time probably you need several hours. But they are going to send you email. And once you open the analysis result window, you can see that there are many tabs here. The first one is the summary. Actually, that's the summary for the pathways, upstream analysis, disease, and function for all the tabs later on. But it usually lists only the top 5 pathways or networks there. What data file? That's the Excel file. Yeah, Excel file having a gene name and fold change. If you need a fold change information, later on I'll talk about how the IPA use the fold change to implement to the figure, to the networks. Yeah, but gene name is the must. And then you click the canonical pathways. Here is the bar graphs showing that. And the orange color indicates this pathway is activated. Actually, the data is the data is comparing the accelerated stage with the normal. So the orange color indicates the phospholipase C signaling is activated in patients relative to the normal people. And the blue color indicates the inhibition of the pathways. And if you need more information, always click this button. It will give you explanation of this, how to, the color, like how to explain the color and p-values and everything. For me, the p-value is not my emphasis here. The p-value is always the relative compared with the IPA database. The z-score I'm talking about later on is also relative to the IPA database. That's like having a Fisher test and other kind of tests. I do have a full file in your hands-on folder. It's called Upstream Regulator. It has a very good explanation of the z-scores and relative p-values over there. Hmm?
So those gray bars and the white bars mean their z-score is 0, when the gray bar means the z-score could not be achieved. What does that mean?
I think that one I cannot remember, just have the more information, yeah. Yeah, it says it's shown here. Yeah, positive z-score is the orange. Yeah, z-score equals to 0 without color, yeah.
Does that mean the IPA database doesn't have any information
No.
to our data?
It has the information, but your data cannot be overlapping with what the information already have in the IPA. Doesn't mean your data is not true. You might have a new discovery. You know, I will talk about the pros and cons later on. It's not for like a novel, very de novo discovery. It's always ask the database. Yeah, don't be nervous if you found a z-score 0 or other things. It's just not found in a database. Yeah, when you click either bars, it will show you the pathway, nice signaling pathway in the cells. And also I have the figure legend here. Yeah, in your handout folder, actually I also had shown the IPA legend file there. You make sure you download it because it also took me some time finding the explanation. Okay. The darker color indicates greater fold change. The green indicates downregulation, and red indicates upregulation. As you can see, this marks really red. It's like upregulated in accelerated CML patients. It's about 8.2-fold. And when you click the networks, it will show you all the networks the IPA got from your data set. The bolded genes indicate that's on your data set. You also have the like red arrows. That means upregulated in your data set. Green, again, that's the same as consistent with what I said before. And those genes with not bolded, actually it's not on your data set. That's the nice thing about the IPA. It can also drag out other molecules which are not in your data sets or not, yeah. So give you a lot of information. And also the IPA has the file telling you how to generate those networks. Usually people pick the top network for their further validation because it's more likely that's involved in your study. Not always true though, but most of the time. And it also tells you the disease and function involved in the network. And the IPA can also export all the information you have. There's an export link here. Okay, when I type the network 1, here's the information. Here's how this picture shows how those molecules or how those genes related, connected to each other. The dashed line usually indicate indirect action. The solid line indicated the direct action. It's shown in your IPA legend file. And also this marks actually, I showed you before in the pathway analysis. It's highly upregulated in accelerated patients. And there's another nice thing is the disease and function. When you click that, it shows you all the disease or functions involved or regulated in the patients. And you can also do the upstream analysis And also, this table also show you the whether this upstream regulator is inhibited in your data set or activated in your data set. And actually, you can always get more information from the file I sent to you. It used to be called transcriptional analysis, but right, it's called upstream analysis because the data show not only the transcription factor, it also show the transcriptional regulator. It can be the transcription factor or growth factor and other receptors. So it's not limited to transcription factors analysis. So here is the mechanical network of the TGF-beta. Yeah, this table shows you that in an accelerated patient, this pathway or the function of the TGF-beta is likely to be inhibited. And this network analysis is showing you how the TGF-beta target those molecules in your data set. Again, the genes having color show it's in your data set. Yeah, you can see the TGF-beta can be directly activated or regulate the molecule or indirect through other molecules. In this example, it's ERB. I think this is HER2. We talk about a lot in earlier lectures. And this is beta estrogen. Okay. And this one show the activated mechanical network. This is, I guess, this fibrosis growth factor 10. This molecule, this transcription regulator, it's activated in an accelerated patient. That makes sense because it's a growth factors, right? And also in IPA, it can show, this figure actually show you the location of those molecules. It can be in the membrane or in the nucleus and cytoplasm or membrane. And there's another feature in the IPA is regulator effects. The regulator effects actually it's the combination of upstream transcription regulation analysis and the disease and function analysis. And the shape kind of changed because when you try to understand the regulator effect, it uses a different tool to visualize the figure. That's Path Designer. They use different shapes for different molecules. Again, it's shown in the legend. And actually, I noticed actually a lot of PIs, they like to use the Path Designer in the IPA to draw to make the figure for their grant application. Because for when you do the grant application, figures are very important. They are eye-catching, so they like to use this one. And there's an advanced feature in IPA. That's advanced. That's why, because it's not obvious on the IPA. But you can do, there's an analysis showing a comparison analysis. You can put the results from different comparisons in the same graph. For example, I put the two comparisons together, put the result of accelerated versus normal or chronic versus normal, and put them together. As you can see, actually, we see the pathway shift. This is disease and function. We show the disease function shifted, pathway shifting. Okay, this shows the different shifting. So it makes sense because there are different stages of patients. used a lot when you try to find the effective dose, because at the low dose, you probably don't see the pathway shift, but when you increase the dose of the drug to treat the patients or cell lines, you can see the pathway shift. Actually, it can provide another evidence to say that dose might be selected by your further study. And now, after giving out that, I would like to give out the examples. This paper actually showed the microRNA 155 affecting the B-cell leukemia and lymphoma tumor formation or growth. In this study, the messenger RNA was isolated from the B-cell, from the microRNA 155 transgenic mice and the wild-type control mice. This is a messenger RNA gene profiling, because the PI tried to find the target of the microRNA 155. And this is the summary of the pathway analysis. I think the top 5 up or down regulated pathways were listed here. The PI actually is studying the B-cell lymphoma, so the B-cell receptor signaling is always important for him, even though that if you look at this one, only like 4 molecules are involved, but this is his main concern, so he's focusing on this pathway. And they found in transgenic mice, the PCL6 was down-regulated. So they used so the next, they extract those genes, downstream targets of the BCL6 and draw the heat map. Unlike the heat maps we talked earlier before, this is kind of biased, just like using a subset of the genes and try to make the heat map to show the change of those target genes. It shows that in the transgenic mice, those downstream targets actually increase. And the IL-6 and CCND1 and c-MYC, they were all upregulated. As you see, they are cancer-related. They are pro-survival genes. So this is one example. The second example is the data I analyzed a couple years ago. The PI actually tried to find the effect of the microRNA to the lung cancer. And he had 2 groups of mice. One is the wild type, another one is knockout. And here is the heat map generated based on the microRNA profiling. And after I uploaded the microRNA into the IPA can also do microRNA, but for microRNA, you cannot do pathway analysis, but you can do network analysis. This is the top network involved in the regulation. And as you see, there are not only microRNAs are shown on this network and other like genes, for example, the KLF7 and P53 also shown on this network. And after we reviewed this network and the PI read several articles, he decided to focus on this gene A. And he wrote a grant, and it was funded, not because of this, but after he decided to focus on these genes, he found a lot of changes in the expression for his data. So So he had a very nice preliminary results after getting focused on this network. So now let's talk about something related to what we had in our workshop. As you remember, Dr. Yu used 4 different methods trying to find genes, top genes, which can predict the overall survival. And as you see that 4 top molecules are common among those 4 methods. But we don't know how they are connected. Now you can use IPA or you can do PubMed search. I uploaded, actually just input the genes in the IPA, then IPA give the network showing how those 4 molecules involved. Very, very nice network. We input 4 genes, but other genes also grab out by the analysis. As you can see that this network actually involves cellular movement and cell-to-cell signaling and cell-to-cell interaction. They're all cancer-related functions. And when I hit the upstream regulator, it shows how those 4 molecules actually related to the beta estradiol, that's estrogen receptor. So that's breast cancer. So we are happy. That's another way to prove actually our method for the statistics has no problem. It's good. And here's my own study. We collaborate with a lot of people in the cancer center and medical center. And this is the collaboration with a professor or medical doctor in a radiation oncology. You know, for those doctors, they are very interested in gene profiling for those DNA repair genes because the radiation, whether the radiation works on people or not, and really relates, not really, a lot of time, they are correlated with the expression level of those DNA repair genes. Because if the repair gene expression is high, the tumor, when you radiate the tumor, cause the DNA break. But if the expression level is too high for those DNA repair genes, the genes got repaired, so it escaped from the apoptosis. So they are very interested in that, in the expression level of those DNA repair genes. So this is GEO example. I downloaded from the GEO website. That's a clinical trial study. It's called JBR10. In this clinical trial, the patients had adjuvant chemotherapy after surgery. They are non-small cell lung cancer patients. There are 2 arms in this study. One is the ACT, the patients having the chemotherapy after surgery. Another group of patients, they just observe. They are all early stage of patients. Right now, it's very difficult to find that cohort because everybody goes to have the chemo or radiation after the surgery. So that it's difficult to find this data set right now. And after downloading the expression data, we actually dichotomized the patients into 2 groups based on the median of the expression levels for each individual DNA among the 240. I asked questions for those 240 DNA repair genes. We found only 12 genes can predict survival for those patients. As you can see that for those patients, I'm going to give just one example. There are 12 of them follow similar pattern. And as you can see for this gene, NUDT1, that when the patient had the low expression of the genes, it survives better. After they have the chemotherapy, it's better than patients having high expression. And also for the observation, like the without treatment patients, the low expression patient have better survival rate. And also we look at the TCGA data. Let's talk about TCGA data. In TCGA data, they have adenocarcinoma patients and also squamous. And they have normal and, sorry, I think I didn't show normal or tumor. But in the tumor, we saw the higher expression of those genes in the tumor samples. So we have 12 DNA repair genes. Now we are writing a discussion. We have trouble because we don't know how we know a little bit about how they connected. So we use IPA to help us to write a good or impressive discussion, even though we say probably a lot of reviewers don't pay too much attention on the discussion. But we just input those 4 DNA, those 12 genes into the IPA, and we did a network analysis. So right now we are writing a discussion based on the connection of those 12 DNA repair genes. Okay, here is the advantage or disadvantage when you're using the IPA. For the pros, you know that it can unlock the insights buried in experimental data, quickly identify relationships, mechanisms, function, and pathways, and finding novel mechanisms in your research. It might not be like a novel in general, but it's novel in your own research. People haven't discovered anything in your research. And another important information is, another usage of this IPA is you can do drug repositioning. Why? Because in the IPA, you can find in your pathway which drugs. You can use the overlay feature in the IPA to see which drug can target the molecules involved in your study. But for the cons, it's knowledge-based, as I said. It's not for de novo discovery. If you are a pioneer genetics, you probably do not like the IPA because you want to find nobody has discovered things, and not for discovering unknown genes. For example, when you see your data, you got a lot of genes having a LOC and number and ORF number and L and C RNA. So the IPA cannot do that, cannot analyze the data. It will just be ignored. Okay, now because I cannot use the computer, we can start to do the self-practice. The gene set we are using today is the comparison results between the cluster 2 and cluster 3 of the TCGA breast cancer. I think you are very familiar with these figures. And the results actually is between those 2 groups in the ER-positive breast cancer patients. So their total genes, 662. One thing I have to mention is that in IPA, the best gene number or optimal gene number, which will be good for the IPA, is less than 300, because you don't want your network too complicated. When it's too complicated, it's hard to explain. So and also it takes a long time. I think the IPA can handle as many as 800, but I suggest you just do less than 500. There's a way to set the threshold, I can go through that with you later on. Just download the data from the, I think from the hands-on, I have a data over there. You can download it and upload into the IPA. Everybody has the IPA